contrary to popular belief, because I don't have a PS5, I do game.
Okay.
Right. We're still trying to figure that out, but eventually that PS5, I'm sure, is just going to appear. Yeah. Antibody design meets AI copilots. Charbel Eid breaks down the future of drug discovery and why scientists aren't going anywhere just yet. Welcome, everyone, to another episode of the Helical Brew. I'm Mike Ouren. I'm here joined with my co-host, Sura, Hadi, and we're super excited to be back. It's been a little bit of a break since we've been on Riverside, and I feel this microphone in front of me feels a little bit new again, which because I had this little tiny thing, while I was on the road in Germany and Hamburg for SLAS, and I did miss doing the live episodes because it was really, these were more field episodes that we recorded, but it was fun. It was testing out the waters on how to do something on the road. But I think our happy place is right here in the studio, and I am super, super excited. Our guest today is going to be Charbel Eid from BigHat Biosciences. He's the vice president of platform, and I cannot wait to explore what that's all about and what BigHat Biosciences is all about on top of that. This is going to go into our AI Thought Leaders series. The impact on AI life sciences has been so prolific already, and we can't wait to see what's coming next.
Oh my gosh, spell that word.
Anyone? I think I read about the spelling bee champion recently, who got second place the year before and got first place this year. So hey, that's definitely a serious player right there. Anyway, I digress. This episode's title is going to be Designing the Future and how AI is reinventing antibody discovery. And Charbel, welcome. Thank you for joining us.
Thank you so much for having me. I'm excited to be here and talk about AI, BigHat, all sorts of things.
So, we always have an icebreaker. we always break the ice with our guests, and we do it in different ways. But it's usually just with a question. And so, in this one, if you could describe the current biotech AI landscape in one metaphor, what would it be and why?
Yeah, I spend a lot of my time these days raising a toddler, and I see a lot of similarities between where AI is and where my child is right now. At the 2 to 3-year age mark, you're seeing a lot of potential, a lot of creativity, a lot of kind of fearlessness that comes out in kids, and I think that's very, very true in AI. I think we're in a phase right now where we're kind of starting out. We're past the very, 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 very first days, and we have a sense of what this could be. And we're seeing signs of potential and a lot of excitement, and maybe soon some maturation, like some growing pains that will come before it takes its final form. It's a very exciting phase. It's a very kind of high promise stage that we're in, but it's one that I think still highlights how much there is left to learn, and before it becomes what it's going to be.
I love that. That's a fantastic parallel, and the other term that's come up in previous episodes has been the hallucinations term as well. It's like, well, there's just a lot of human connections and human development, as well as things that can happen in a state of mind that's maybe not on the rails, as they say, and so.
Yeah.
But I like the analogy because it is a living
definitely
system, and it demands care,
has
cooperation, kind of a long-term view like children do.
Yeah, it's very interactive for sure.
Yeah, what you put in, you're going to see the results in that direction. Maybe not exact, but it's going to influences are key in AI, and that's a fascinating element in technology for me personally, because it's always been in the technology sector. You have a fixed input and a fixed output, and there's obviously a lot of decision making happening in between, especially in control systems, but it doesn't have the adaptation capability, or hasn't historically, and we haven't thought of technology in that sense. And the fact that we can now look at technology with a human eye is pretty fascinating. So it's pretty cool. Well, I'm looking forward to doing a deeper dive and just a little bit of background. I love to kind of explore how you arrived where you are, but I'm just going to give our listeners and viewers a little bit of a background that you are the vice president of platform at BigHat Biosciences, and you lead the integration of machine learning and synthetic biology to transform how next generation antibody therapeutics are discovered and developed. So, but I'd love to kind of learn a little bit about how you arrived at where you are today.
Yeah, absolutely. So I would say from a young age, I've had the personality of someone who's very interested in data trends and kind of seeing what I can get out of very random data. I remember when I was a young lad, like kind of trying to build data sets in Microsoft Access, which I don't even know if that's still is a thing still.
I remember. I remember.
Around like TV viewership ratings and things, random things like that, where anywhere I can mine some insight. And I've always been interested in that. And I kind of did my PhD in Stanford here in the Bay Area, focusing on microfluidics and assay development and that integration between, or the interface, sorry, between engineering and science, and how to apply scientific concepts and principles in a more engineering system than the more traditional pure biology or chemistry around me. And then went into automation in my first industry job in a startup, where the goal really was to build integrated systems with LIMS components that were minimizing and kind of streamlining operations, capturing all the data and metadata, executing scientific hypotheses in a reliable, repeatable way. Today, it turns out we were doing was essentially building the lab portion of an AI lab, right? And so that was an early exposure that I had in my first job into that world. And also a startup, which was very helpful to kind of see the fast pace, the errors that happen, the risks that are taken. And after a couple years in that job, went to Gardent, where I worked briefly with Sura as well. And there it's a larger company, more advanced, focusing on building liquid biopsy tests for screening and monitoring and detecting cancer. And that was a very, very kind of pivotal experience, because you get to see kind of the responsible side, right, of the technology of biotech, right? When you're dealing with patients, with the FDA, when there's a lot of stakes, when you're being regulated, and making sure that you're innovating in a highly responsible and somewhat constrained way, rightfully so. And it was a helpful mix of the two worlds, between kind of a startup environment and then this larger company. And then more recently and currently, I'm at BigHat. I joined the company when we were about 10 people. And there, I joined early on. Like, we were in a incubator out here in the Bay. We had like a bench and a half. And we had the goal, the vision of building a platform, an antibody therapeutics platform that was automated and integrating synthetic biology and machine learning. And it's really exciting, I think 4 years on, to see how far we've gone and how big our platform is, how advanced our therapeutics are, and that we're getting closer and closer to helping patients. We're focused on building better medicines, better biologics, in a faster, more cost-efficient, and more kind of addressing problems that were unsolvable before. So you can think of traditional monoclonal antibodies. These are the types of antibody therapeutics that a lot of us know. They've been around for decades, but they're limited in what they can do. They're constrained in the types of targets they can go after, the kinds of diseases they can cure, and that gave rise to what are known as next generation antibodies. These are more kind of designer antibodies that you can think of them as, sometimes they're fragments of a traditional monoclonal, sometimes they're smaller. They could be more complicated, bispecific kind of molecules that can go after multiple targets at the same time. The thing about them is that they're much more powerful and have higher kind of potential to go after targets and diseases that were previously kind of undruggable, as we say in the field. But the challenge with them is that they are harder to synthesize, to make, to develop using traditional antibody therapeutics approaches. They're ill-suited for the job at hand. And so when you're making a therapeutic, you're trying to optimize multiple properties at the same time. You need it to do a lot of things, right? You need to bind its target, right? You need to make sure it actually reacts with the thing you want it to react. It needs to be functional. It has to be stable. It has to be pure. You have to be able to manufacture. There's a lot of things that have to come together for it to be a good therapeutic. And if you think about kind of antibodies or proteins, the search space is just massive. Like every single position, you have 20 options, or there's 20 amino acids, and that's just keeping with the standard amino acids. And so you can imagine how that blows up very, very quickly. And so the combination of the potential of these class of molecules, the difficulty in using traditional approaches to actually kind of achieve them or develop meaningful therapeutics, and just the breadth of the search space, the parameters you're trying to optimize, is a great combination, right? For that justifies and kind of really gives rise to combining high throughput experimentation, synthetic biology with machine learning and artificial intelligence to kind of help whittle the space and kind of guide you in the right directions to kind of achieve multiple objectives and optimize molecules in a kind of high quality but efficient way. And that's BigHat. That mission was existing from the start. It really appealed to me when I heard it. That didn't really exist in the lab or in the company, but was a kind of gleam in our co-founders' eyes, and our founders' eyes. And it kind of appealed to me to get to join early on.
Really
I can
putting on,
tell.
yeah,
You're very excited.
yeah, very passionate. I love it. Yeah, I love it.
It's-
And really putting on the big hat to go after some really big problems. I mean, these non-trivial, you know.
We take our hats very seriously. All our projects are after hats.
You can't go wrong with a name
really?
like, you know.
Milliner, someone who makes hats.
That's
So
right.
That's the platform making hats. It's we're pretty committed to this.
And that really is, I couldn't think of a better segue into our first segment of the show on how AI powers biotech innovation. And, you know, give us an idea of what the Milliner platform is all about and how that works and the machine learning aspect of it.
Absolutely.
And you've given us a nice background already to
Oh,
kind
yeah.
of the bigger picture, which is really nice for folks who may not be familiar with the space.
And before you start, I do want to point out that is a fantastic term. I had not heard it before. Undruggable. Because that is like next level. So it's you were going after undruggable stuff because that's, you know, everybody like, well, I mean, the mindset of the average consumer. I'm a roboticist by trade. I'm not going to go into my background, but you know, for me, there's a lot of novelty in what I hear sometimes, and that's just the keywords that come up sometimes. It's just fantastic. That's a great one. I love that. Undruggable.
I'm glad. I can't take credit for it, but it's
okay. I'm sure. Yeah.
Trademarked by Charbel.
Take it anyway.
They're not on this podcast.
That's true. That's true. Yeah.
But as you said, a Milliner makes hats, but in this case, it's a platform to do what you all need to do.
Yeah, that's exactly right. So I think the background is helpful, hopefully, to kind of lay the foundation.
Fantastic.
We're going after difficult problems, and we're trying to make antibody therapeutics that have to meet multiple criteria. One of the core principles of our company, like we've built the platform to solve this problem. And what that looks like in practice is we have a multifunctional / multi-arm platform that does a lot of things. The core of the platform is this design, build, test, learn loop, which you hear a lot about, especially in machine learning context. But just to say like what that is, it's you're designing a set of sequences or variants. Like you make a mutation in a protein here at this position or that position. You build them. You actually like make the proteins, test different properties of them, the things you care. You run assays, right? That's what it breaks down to in our scientific world. You get that data, then you feed that back into your machine learning models to learn from it and then propose the next set of variants, right? So you're iterating on, you're learning from N, and then you use that to design N+1. This is really, really critical. And again, we've built our platform around making this cycle and this loop really fast, really robust, very high throughput, and very, very high quality. So to do that, we focus on a lot of different technologies that make that possible. We use what's called cell-free protein synthesis, which is a way to take DNA and express it and translate it into proteins within a day. It's a plate-friendly format, so it's a high throughput automated format that allows you to go from kind of start to protein very quickly. We use automation across the board in our assays. We've built a suite of over a couple dozen different high throughput assays that assess function, binding, thermostability, other types of stability, purity, et cetera. Cell-based functional assays, all sorts of different things you might care about when you're making an antibody therapeutic. And we've also invested kind of equally heavily in the what a computational layer. So all of this is underpinned by a custom built, we call it LIMS++ system that captures everything that we do, from the sequence design to the lab workflows to all the QC pipelines and all the machine learning models and training and all the metadata. It's all living in that simple shared space, that common language that we have across the company. And so in practice, the platform looks like just a whirring engine. Like it's always running. We're always making proteins. We're always expressing and characterizing them. We focused a lot on modularity, so we're able to support many different therapeutic programs simultaneously, over 10, 20, essentially as many as we want. And we've organized ourselves around this ability to iteratively optimize proteins. That's ultimately like the thing we believe in, that consistency and speed and iteration is the way to address these highly complex multi-parameter problems. Machine learning exists across the board. It's the main engine that we use to design our sequences, our variants. It's the guides and fuels our QC as well. And it's kind of ever present. But importantly as well, like to do this iterative optimization, you need to start with something, right? You need to have a molecule that has some property, some signal for you to kind of improve and optimize upon. And in that area, we've focused a lot on flexibility. So this is what's called, what I'm describing the core work cells, kind of the optimization phase. There's also like a discovery arm that allows you to even start with something that you can then optimize. Because we have such a strong operational scientific core work cell, we're able to be very flexible on what goes in, like the starter material. And you can imagine an alternative world where this didn't exist, where we didn't have this core work cell, where we put all our eggs in the discovery basket. Like you have to use some approach to go from 0 to 100 with just discovery. And that used to be a model that was valid for easier targets, the older days of drug development.
Lower hanging fruit.
Lower hanging fruit, exactly. But at BigHat and where we are today, that's not really valid anymore.
It's all been picked.
Branches have been picked. Right? We're not climbing up higher and higher. Our discovery focus is very flexible. So we can start with anything. We have internal efforts around using display kind of technologies that allow you to pan your target against a whole bunch of different sequences, billions, just to see if there's any that have any signal at all. We have machine learning based approaches, AI based approaches around kind of using structure of the target and potential antibodies and kind of see what might computationally in a simulation fit, and then testing that in the lab. We also are, sometimes we have in our partner programs, the partner might have a sequence or set of antibodies that they're interested in that have some of the properties they care about, but not all of them. Essentially, it doesn't matter. Like we recognize how kind of modularity is important here. And as long as we have some starter sequence material, we're able to apply the full suite of capabilities on our platform to kind of optimize very quickly and go from something that maybe binds well but is unstable, or is pure but doesn't have good function, to molecules that can meet the broad spectrum of what's needed.
So BigHat's not just mining, right? Or using like what's out there and compiling and informing and using the model to really kind of suggest. There's a wet lab component, and all that data feeds right back in. So from start to finish, that is the platform?
Yes, exactly. That is exactly right. And that's a good question, actually, because it's a good point, sorry, because we would love to use the data that's out there, and we do. There's useful public data sets. There's useful kind of external data generation companies as well, like CROs, et cetera. And one of the central hypotheses of our company is that data generation, like highly bespoke, well-designed, fit-for-purpose data generation, is the thing that drives AI forward. That's the thing that differentiates a drug therapeutic pipeline and project from another, not just a good model. And so that's been why we've focused so much on high throughput wet lab platform that integrates with AI, ML, with what's out there with public data sets, et cetera, but that has a very strong internal data generation on it.
Very interesting.
So you're really using this as in a way, a closed loop process within your organization to drive the next experiment using these data sets, right? I mean,
Totally. Yeah,
you're,
absolutely.
and with the AI in between, right? But one of the other questions we had was like, when you take all of that, how do you balance the speed, the accuracy, the safety, all the things? Because now you've got a power tool at your disposal, integrated into your workflows, integrated internally. And I love to hear that you've really, I mean, sounds like very thoroughly embedded this into your organization, your process. And so, but how do you balance that out when you're trying to run things and
Yeah,
using the AI?
it's it's a kind of multi-layered approach, I would say. To some extent, like one of the benefits of using AI is that it, especially at the early phase when you're looking at this massive sequence world universe that you have to explore, it's a wonderful tool to explore it efficiently and kind of smartly. And so that it gives you at the discovery phase of drug development a big speed advantage. Now, at the same time, as you said, like, you have to be careful about what you're doing here. There's a natural layer of safeguards, which is if you design junk, the protein won't express. Like there's a scientific layer, like things will just fail,
Just doesn't work.
which helps. And that's easy. That's the easy one, right? That's a kind of easy to catch. Then maybe the more pernicious, like concerning path here would be if you have something that's harder to detect in the bleeds that you're optimizing, something that's toxic or immunogenic that you're unable to maybe see, that looks like behaves well, kind of has the right properties at the screening phase. There, I think it's really there's kind of being a responsible therapeutic company. And also maybe worth keeping in mind the broader kind of pipeline. Like ultimately, if you really zoom out and you think about going from, I have an idea, like I want to develop a therapeutic against this target or this disease, all the way through, I'm going to put this in humans, there's a lot there. And this early discovery phase ultimately is relatively short in what it is. And it's really kind of, it's on us and on the FDA and the broader space to be very responsible with the steps that the phase gates that go along the way. So for us, for example, like any signal that we get, any promising leads that we find are immediately validated in more mammalian-like systems, like things that will give you a hint of something kind of off with what you're doing. But there's also small animal studies, there's tissue studies, et cetera. All of these, I think, are critical. They've been critical from the start, AI or not, to ensure that what you're building, what you're designing is safe and efficacious. And I think the fact that it's AI designed, in my mind, doesn't change that. Like the fact that you're moving quickly, yeah, is great at the outset, but you're still going to do the same responsible studies that we as an industry and as a field have agreed are necessary to convince ourselves, regulatory bodies, stakeholders, patients that the molecule is effective and safe. So it's a natural balance, I think. And it's really behooves us as a company to know any problems with our molecules as soon as possible, right? Because so we can pivot and adjust. So we're all very incentivized to do that.
But see, what you're saying is that the downstream really doesn't change. It's just you don't have one PhD in one room going through all this data and trying to figure out how to design proteins. You have a model that's just generating them quickly. So that part is quick, but the same kind of steps and phase gates still apply.
Absolutely. It's kind of like going really fast to secure your spot in a traffic jam, right? Like really fast, and then you get to the bottleneck.
And then you see people walking by you.
Yeah, that's it's the bulk of it is downstream. Like it's quote unquote downstream, preclinical studies, clinical studies. Like, and there's a world in which AI can help accelerate and improve and make that more efficient. But that's not where we are today. I think today we're very much on the early phase.
And you've probably built those parameters into your model as well, right? You don't have to actually get to the point that the protein's not expressing. Before that, the model will tell you, theoretically this could work, but technically it won't.
We continuously improve our models to account for more properties. So in terms of expression, absolutely. Like it's one of the things we're increasingly able to predict, that this kind of mutation will ablate expression and you're going to lose your molecule.
Yeah.
There's more, I think, where we're trying to go and where it gets harder, and I think that's there's a lot of opportunity, is, as you go downstream, in the process. A lot of things changed. You can think of at the early phase, what we're working in 96, 384, 1536 plates. Like we're doing high throughput, small scale. Oftentimes, you're also looking at a slightly different kind of format of the antibody at the screening phase than what it looks like at the end, when it becomes a drug. And so there's all these types of differences that create what could be a translatability gap, right? As you move forward down the pipeline, you're moving down to a smaller N, higher scale, more drug-like studies. When you're starting to put it in animals to see in its kind of final format, how efficacious is it, how stable is it? When you make it at much, much larger amounts than you are in the lab, things can change there. And there's a lot of opportunity and potential in trying to predict those things, like those assays, those properties as upstream as possible. And it's hard. It's a hard thing because the downstream, you have much less N. So it's less well suited today for machine learning kind of based approaches because you're lacking enough data today. But if you can predict some of these things upstream, you can de-risk a lot of your own projects later on. And that's where we're investing a lot. One of the core questions that is asked in therapeutics is what's called immunogenicity. Like, is this going to be immunogenic or kind of harmful to humans in kind of that immune environment? That's harder to know when you're operating in a cell-based system or a cell-free system or even other kind of species. And so there's a lot of work on this. There's a lot of folks are building assays to try to predict this. There's no holy grail yet. This is one of those things where if you could do this well, you solve a lot of problems. We're working on that as well. And that's the kind of thing where you could use AI to increase almost the safety of downstream steps once you get it right, once you crack the nuts.
It's like our last guest said, right? AI will give you a recipe to make spaghetti using gasoline, but that's not necessarily what you want.
It's got to bounce off the walls a little bit more to hone in on. We just want to stay within what's edible, you know. That's
I could see
all
my
we
toddler
wanted.
trying to make
Yeah.
some glue.
Can I try this styrofoam instead of meatballs?
It looks fine, right?
Sure, yeah.
Be creative, right? There's a lot of, I think in other areas I come from the mechatronics world, and in my head I was drawing a lot of parallels to what you were talking about in terms of, it's a little bit compartmentalized, right? Software with not very good hardware to control the motors doesn't, spending a lot of time to improve that software doesn't make a ton of sense. Versus, if you've got motion control gets better as the electronics get better generally. Motor technology is one of those things out there, that's kind of like manufacturing your therapeutic, right? That's sort of a thing that moves even slower than the actual approval process, right? But then the next slowest thing is the hardware behind, next to the motor. And the thing you can do on a whim is all the software, which I parallel that to research. And right now, most of the changes in software and motion control are because there are prolific changes in the hardware, miniaturization or frequency, control loops, all those things. But it's very interesting to hear that. And it makes a lot of sense, right? I mean, when the research has a proven line, then the clinical can begin to learn from that, right? And then when you get to manufacturing, you can learn from that yet further. And but if you were to boil it all up into a big challenge that you see, right now, I'm sure this is sort of a dynamic thing, but what are the biggest challenges you see in scaling AI molecules into a clinical trial? What is that? Because we already talked about the bottleneck, right? Rushing to sit in traffic, right? But I mean, what do you see as the main challenges for that?
Yeah, I think it is this gap that exists between where AI is being used today and then what matters in a clinical trial. I think that bridging the gap as much as possible, being able to predict downstream modes of failure and properties at scale and final formats, all the kind of things that matter more exclusively more easily, I guess, to assess in a drug-like format. As much as you can do those upstream, I think that is, to me, one of the kind of the biggest challenges in translating the AI focus and discovery and optimization down to the clinical trial kind of development phase. It's also, I think, important to note that this is early days for this kind of heavier usage of AI in drug development, and there's a lot to see yet. We haven't getting this first wave of FDA approved AI designed molecules will be very transformative and unlocking for confidence, for kind of what approaches work. There's a lot to learn yet.
That was what I was going to ask. Is the FDA even ready to consider AI designed molecules into clinical trials, right?
Well, ultimately, if you're doing the right studies, upstream, it kind of doesn't matter how you got there. If you can get there by someone just random number generator and like making mutations that work, or you can use AI, as long as you're proving that your molecule's working safe, you have the right kind of package for the FDA, it kind of doesn't matter, right?
I guess it takes one bad actor for the FDA to get more stringent
100%.
on how you discovered it. Yeah.
100%. There's definitely that possibility. The rigor could increase. I think that's maybe sometimes a good thing, sometimes a bad thing. Like there's a lot of levers that the FDA could pull, and that does interact with how companies like BigHat and those like us who are on the kind of the frontier of this new area behave, and kind of the responsibility that we show as responsible kind of entrants into this field.
Every time you say responsible, I want to put quotes on it. I don't know why.
I hope not.
I hope that's not a quote
Not
state.
for you, but when you were talking earlier.
What's happened that responsible needs to be in quotes? Oh my God. Yeah, that's funny. Well, and then what you say about the FDA makes a ton of sense, right? They're just you're generating data at the end of the day, and you're gonna present it in the same form you did previously. It's just where that data is coming from. Now the key is, and we saw this in the surgical robotics space where I worked in, is don't say that the surgical robot is going to do it better than anything.
Avoid
Never
better.
say that.
Yeah.
And I'm sure somebody is not going to go, well, I hopefully not going to go, well, this AI generated thing, and that's like the title of your submission. No,
Yeah,
don't do
not a
that.
good idea.
That's
Yeah.
not that's not how it works. Just
Yeah.
Don't
let it
flag
do
it.
its thing and present the same document just with better things behind it. Yeah, it makes a lot of sense. And I got to imagine partnerships. I understand you're partnering with Eli Lilly,
Yeah.
right? And how do the partnerships shape your innovation? Does that kind of help you get to bounce things off each other? And
Yeah, they're really powerful, I'd say. They've been a very meaningful aspect of our company. These partnerships with established biopharma and other biotechs. Eli Lilly is as a wonderful example. They've been very powerful for us. Like, we're a small, nimble, technologically cutting edge company, right? Like, we're working on this frontier, but there's a lot we don't know. Like, we're learning a lot. We're a young company. A company like Eli Lilly has just tremendous scientific expertise. They know target biology well. Like, they know the targets as well. They know what patients want, need. They know what diseases and targets are kind of really moving the needle. They have a lot of built-in expertise there. And just in general, they have a ton of know-how. Like, they've been a leading biopharma for decades across different iterations of pharma and biotech. They operate in different spaces, especially in times like today where there's rapid evolutions all around us. It's these types of partnerships are really a great way to innovate, but stay rooted in real world problems, real world patient needs and solutions. And we're working on problems that they have, like real targets that they're going after, that they care about, that they're challenging because, as we said, the low hanging fruit is gone. And so they benefit from different ways to approach these problems. We benefit from their expertise, and also both of us benefit from the de-risking effect that exists. We're sharing the risk in this working out, and we share the benefit in it too. It's a very good driver for innovation, right? When that's the case. And then there's generally, like, what I would say, a bit of a flywheel effect. Like, for our platform, we learn from these partnerships, and we get a sense for what works, what doesn't work. There's a lot of benefit for us to make us better at innovating as well. It's been existing for a long time, this kind of model of large pharma companies working with biotechs. And I think it's a very healthy and kind of symbiotic relationship. We are seeing it firsthand at BigHat. We've benefited a lot from it, and a lot of great stuff has come out of these partnerships with companies like Lilly and others.
that's fantastic to hear. And I hear more and more about collaboration in this industry than ever. And I feel like it's just, yeah, you really have to collaborate. And I can only imagine how many therapeutic trajectories that Lilly's been on in their long history, right?
Yeah.
that we hear about the pharma side of it, what's released into the market, the
Yeah.
trade names of different drugs, we hear about that, but I can only imagine to arrive at the number of products they sell, how many things they've tried to solve, and now they can start pulling a lot of that off the shelf and
Yeah.
re-looking,
They're so good at that. It's a wonderful thing. Like the ability to know what data you need to stop something is more valuable, honestly,
Yeah. Huge.
than even getting it done. Like it's so valuable to have that killer experiment, as they call it, as early as possible. And you build that muscle, right? The reps that you get as a large company operating in many different programs across such a long period of time, that you know how to do this. And that's a wonderful skill that, as a biotech, you have to learn, right? Like you're betting a lot on a handful of programs. Ultimately, you don't have the funding to do 1,000 different programs.
Yeah.
And so,
And that's what we were curious about, because that partnership should shape the innovation pipelines, not just innovation, because then you have this access that you wouldn't have had, because you can start right from where they left
yeah,
off, basically.
yeah, exactly. Like I think one of the ways that this materializes a lot of times, that one of the risky parts of taking on a therapeutic program is to ensure that, for example, like the biology is there, the target is a good target to even go after. And that's not a purely scientific question. There's also the business risk, the clinical need, who else is operating in that space. Those are key questions. A lot of times our partnerships exist in a world where that problem's solved, right? And we're able to focus on kind of scientific innovation within the realm of a well-validated target and problem.
I love that.
Yeah, and it's great. I think it allows us to focus on kind of moving forward in a way. Yeah.
And driving that focus, obviously for kind of what you're going to go after, but just switching gears a little bit, can we talk a little bit about the future of AI-driven drug discovery and therapeutic discovery? Where is there anything that stands out as super exciting in the near term and maybe even long term? I don't know how long term you can get in this space, because it's
That's true.
Exciting tool.
what somebody called long term is like, well, that's only 6 months.
That is incredibly true.
For AI, it's probably the next month.
It is remarkably true how fast things are moving. Yeah, I think there's a lot of exciting things on both the wet lab part of this AI lab and on the computational end. There's a wonderful amount of work that's existing on open source structure prediction. People might maybe know about AlphaFold and the recent iterations of that. And those are wonderful tools. They help a lot in kind of getting started, getting an understanding of the landscape. And also another instance, like recently last year, there's David Baker was given a Nobel Prize, and his lab in University of Washington does wonderful work in what's called kind of de novo simulations, trying to predict antibodies that bind to different targets. And that stuff's open source as well. And these are wonderful tools because they're easy to access and use, and they're kind of a great complement and benchmarks to work that a company could do themselves. It also opens up the space to others to kind of build upon and iterate and innovate upon. And on the lab side, I've been interested and happy to see push forward in some of the automation infrastructure that we're seeing. I'm happy to see more kind of modular approaches to integrated work cells. I think that's been a long time needed. And at BigHat, like, we especially appreciate that because we built around modularity. Like one of the core ideas that we have is that the space that we're operating in is just moving super fast, right? And that's true in the lab. Like, there's new technologies coming out all the time, new assays, new capabilities, and then even more so on the computational end. Like, there's new models, new data sets, new features that are coming out all the time. And so our platform is built around being modular, like being able to plug in different things as they come up. So we're not focused on like our internal effort to do this or that. Like, we're positioned to benefit from what others around us are innovating in and making public. And it's helpful. Like, that's been often a problem with the capital expenditure side, like the hardware side of labs, especially with automation, like these big bulky systems. And so it's great to see more modular work cells coming out that you can plug in different instruments. You can change it around as needed, at least a little bit more so than before. And on the same end, I think there's been a rise of companies that are more kind of the universal driver, I would say, of automation, trying to abstract away working with a Hamilton or TCAN or other kind of systems into a more shared language like Python or otherwise. I mean, it's in early days. Like there's a lot of companies trying to do this. I think adoption still takes some time. It's long-standing players here, but you can see the ground moving a bit, and it's really great because it abstracts away one of the big bottlenecks of connecting computation with lab execution. And I'm very happy to see kind of that movement in that direction. So there's a lot of tooling around. As you said, like if we talk a year from now, it's going to look different from it is today. I'll have a completely different answer. But I'm very encouraged by trends on both computation and lab sides.
Yeah, I'm so happy to see more and more compartmentalization and modularity in software. Now, like you said, it's not all there. It's not all, we don't, there are still some things where you got to do a hackathon to integrate it. Hey, everybody, let's do a hackathon. Let's integrate this instrument into this system. But that's just those exercises ultimately, and Sir Welt knows this more than anyone, they make you better, they make you smarter and more agile for the next hackathon, because you can
absolutely hackathons are great. Nothing wrong with hackathons.
Owning your automation, owning your platform is one of the most prolific things I've seen over the years in the new companies like yours, and just bringing that all together. So it's really great to hear that. And you're going to have to work around some stuff. Some things it's like, well, I can only use this one language on this one instrument and bring it together. And I feel like also the modularity was maybe 15 years ahead of its time in where it's useful, but now it's truly useful.
It's necessary.
Yeah, you can't go without it because the dynamics of your experimentation, you need to be able to move things around on the board like chess. I mean, you just, Yeah. So really.
Well said. I remember trying to build those back like when automation first started and people just wanted systems that stayed. And I was like, but you can dock a table. Don't you want to move it?
This is one thing,
It's just one thing.
Yeah. You got a lot of paperweights, I think, when you can change things around. Yeah.
I mean, do you feel like the AI could be almost like, so let's just a little background, Copilot, Siri, these products that consumers are mostly exposed to, there's a lot of consolidation and unification behind the scene. Like Siri, I know Apple's approach to AI integration has been one of partnering and using back-end engines that we all know, but just making them really easy to access through their one interface. Do you see that maybe as a future co-pilot for biologists and
like a little DNA that says, I see you're designing a protein.
Oh, the paperclip. I love it. Yeah.
Certainly, I don't see a reason why not. The pace of change in AI in biotech has been so fast, like even a couple years. So not going to predict any kind of cap upset here. I think it's the what's the world is our oyster or something. I think it's already the case that you could use an AI. You could use AI as a copilot for your thought process and your design kind of approaches and data interpretation. These things are there. There's increasingly like a wide number of AI scientists or AI bioinformaticians, and again, it's all early days. Like none of that is where it's going to be. But the systems and the components of a world where AI is sitting next to you in terms of solving difficult scientific problems, it's there. Like the components are coming together, and over time, it might take a little bit of time, they're going to get there. The fact that we're operating with like multimodal, multifactorial systems, like it's a great opportunity to actually benefit from AI to sift through complex data, to help you kind of reinforce the scientific expertise that we as humans have. And so I'm very bullish on that kind of trajectory.
Yeah, that leads to a bold prediction.
Get ready to be bold.
And then I was just thinking about it, sir. This is maybe the segment where if I could just get a little bit of rights, just tad to the Star Trek theme, this is what I would play right now. Because, and this is really, 5 to 10 years. 5 to 10. You got some connection. So 5 to 10 years, is a really long time in computing. It's a long time these days in Even robotics, it's maybe not such a long time in robotics, but in software and biotech in general, 5-10 years is a huge
It's a
amount
long
of time.
time.
It's a long time. I mean, in all fairness, that's why I say Star Trek's worth, in a galaxy far, far away, Star Wars, pick one. But, do you have a bold prediction in biotech?
I mean, I
Could be
think
anything.
actually a little bit. My bold prediction might be a little bit of a different track from what I've been saying. I suspect that there's a lot of hype right now, and it's early days, right? There's a lot
Hype?
of
No way.
I know, unheard of, right?
Say it's not.
I kind of I think there's a lot of claims. It's true in AI in general, but in biotech, a lot of maybe hope that using a model-only approach would be sufficient, that you can go from idea to drug, with minimal, I don't know, lab validation. That's kind of the appetite in some spaces of the market. And I predict that we're going to hit a point over the next few years where some of these things are not going to work out. Some of these bets are going to fall and fall flat. And that might build some skepticism about the use of AI in biotech. I think that could be where we end up. Like a few years from now, we're in a more negative space in terms of how we think about the utility of AI in biotech. But that underneath of that is the technology will continue to improve and improve significantly, and a lot of great work will happen, and then it will take off. I can see some trouble, like turbulence, before kind of landing the plane. And I think that the best AI companies will be the ones that have the best data generation and the best data, not necessarily the ones with the best models. And I think that will work out over time.
What's that saying? All good stories have 3 acts. That will be Act 2.
Act 2.
And Act 3 will be the the great rise, the triumphant of ta-da! We've resolved, and and then there'll be sequel over and over again, I'm sure, and continuous development. But no, it's awesome.
Well, every character needs a conflict before they grow.
That's right.
Yes.
That's right.
Yes.
And every technology does too. I mean, ultimately, if you look at the history of computing and software, there were always a lot of just Windows 98, Windows 2000 is a great example of where that was Act 2. This is too much. Time to.
I was already traumatized when you said 98.
Yeah, right. Yeah. 98 was the end of Act 1, and 2000 was just okay. Here we go.
You're dating yourself. Actually,
I know.
We all are. Yeah.
I know. Yeah. 68,000 Macintoshes. So don't get me started. Yeah, that's I mean, it's pretty awesome what you're doing, and the fact that you've got those collaborations, like you said, sharing the risk and bringing that together with companies that have so much background on trying different things out, right? Because they've been around for a long time. And
absolutely.
if you wouldn't mind switching gears a little bit to career, I think there's
AI
a lot
thought
of
leader.
AI thought leaders, right? I mean, and there's a lot of aspiring folks out there that want to get involved in the industry. But do you have some key moments that shaped your transition into AI? I mean, it certainly wasn't something we all went to school for.
Especially in this leadership role, right? That you're determining. Yeah,
absolutely.
I think I almost got fortunate. I would say, as I mentioned, as a kid, I had this kind of pre-wiring, I think, for this kind of world. And I got fortunate almost with my first real industry job being one where you're building integrated systems and capturing all the metadata and data and systems speaking to each other. Because that really, that at the time was the lab portion of an AI lab when AI didn't exist. And then as the models and the artificial intelligence itself kind of improved significantly over the past few years, I personally had this appreciation for the value of generating kind of high quality, high throughput data. And so then I think having that technical background and personality kind of pre-wiring, with seeing the rise of AI and machine learning as a potential tool even a few years ago, and getting the opportunity with BigHat, to build a platform with this philosophy at heart, when we mold it into a lab that's made for AI, that's made for ML, where every component is designed to streamline data back and forth to a LIMS layer, to incorporate validation, like to go from machine learning designed sequence to like molecule in the lab. Like all of these layers are the bridges are exist between every kind of computational and lab layer. That opportunity, like at the time that I was looking for it, was also very fortunate. And I think that itself, like building this lab for AI and then seeing AI rise, and kind of being able to adjust how our platform operates, to keep up, to stay ahead, to kind of shape how AI is used in biotech. That's been kind of a defining part of my life, frankly. And it was built on little things before that, like getting the right experiences in schooling, getting the right kind of job that kind of put that kind of thinking in my head as well.
You're gonna have so many people apply to BigHat after that.
uh, yeah,
A lab with AI in mind. That's awesome.
Yeah, it was less. Like when we started, it was less obvious that this was the path forward. This is kind of one of the cool things. Like we were taking a big risk, I think, in building the lab this way, because you could
At the
kind of
time, for sure. Yeah.
yeah, and now it's very common. I think now it's more accepted as like the way forward. But yeah, it was good.
It's not, yeah. It's just I feel like you really connected with the vision of the company that you
Exactly.
joined.
That's
You
exactly.
just, it was. I must apply.
Yeah.
Yeah, that's awesome.
Yeah.
That's awesome. And as far as like for young people who have aspirations of doing exactly that, right? They could hear that presentation and feel like, yes, that connects with me, or other companies that are coming up. Any advice that you'd have for folks that want to break into?
Yeah,
biotech
I'm very sympathetic. Like it's a very tricky time, right, to be a young scientist, young engineer when so much is changing. What I would say is not to over-index on AI per se, partially because it's going to change a lot, and partially because it's a bit ephemeral. And like, what does that really mean? I would say the key thing here, like maybe two key things, like identify the differentiating skills that you have, like whether it's being an automation engineer, whether it's a protein scientist, a machine learning scientist, whatever it is. Because those skills are the thing that you will use to make immediate value, and they're going to be very useful for a very long time. Like we're not going to move away from the utility of those skill sets. At the same time, the labs of today are integrated labs. Like that's where we're headed. Like we're, I think, past the point where there's the throw it over the fence kind of approach to tackling difficult projects, whether it's in therapeutics or diagnostics or any part of life sciences. As a scientist, you need to understand a little bit about AI and ML and kind of what goes into it, how scientific data interacts with it. You need to understand like the robotics and at least from a general sense of how they operate, what matters, what doesn't matter. And you can make the same analogies if you're starting from a computational side. I think it's important for the young scientists and engineers of today to have fluency, at least like basic fluency, in all the different components of a modern lab. That will help you amplify your existing differentiating skill, and it will give you a chance to adjust, and kind of keep up with the shifting winds and tides of whatever is going to come next. Because it's a long ride. And you need to have the skills to be able to like navigate that, rather than just be the best like a protein scientist you could be with no interest in other things and hope that that's going to be sufficient. I think it's not the best approach today.
Or semi-young who want to pivot.
The same, yeah, that's true for everyone. Like I think broad fluency is really critical today. While underpinned by the thing you're really good at, or the couple things that you're really good at that will add value immediately.
I like that.
And I'll throw this one in here, but I think what you just said is so critical for folks to kind of guide their career if they have interest in it. But just flipping it over to the industry side to make things smoother, is there a mindset shift do you think the industry needs in general? There's always a mindset shift as industries evolve. I've seen it in the robotics space.
Yeah.
I'm curious what your thoughts are in this space. Is there something that we're trying to do the old way, maybe too much, and we just need to, shift into a new gear?
Yeah, I kind of see that as the shift here. Like there's I almost see like sometimes I observe what could be described as a bit of a tension between the traditional like scientist only led approach to tackling difficult scientific problems where the expertise lives in someone's brain or a couple people's brain, and there's this tried and true way to kind of resolve difficult challenges, kind of optimize properties one at a time, et cetera. There's all sorts of like heuristics around that. Well, then on the other hand, the model will solve all kind of thinking. Like if we just build the right model, everything will be solved.
One thing.
And I think
Or
that,
feed the model.
yeah, exactly.
Right.
Yeah. And I feel like that whole tension is the shift that we need to move away from. I think we need to like really understand and embrace the way that AI can supplement scientific expertise, not replace it and not do without it. Like we're going to run into problems and it's not going to be perfect. And it's like there's a natural, understandable desire to raise firewalls among like existing players and to try to like fully disregard the way that things were done by new players. But I think the right shift here is to figure out the best ways to leverage scientific expertise to make our AI ML models better. We've done like one very minor example that I really like in our company is we're doing very complicated assays that are hard to interpret. And we've built QC to try to like understand whether this curve is good or not. And it's very minor stuff. Like for some of the more complicated assays, you're thinking of it as little as like, for these 5 seconds, this curve went up instead of being flat. Like, what does that mean? And we have SMEs who are wonderful and really strong at understanding and interpreting this data. And we have kind of ML-based QC as well. And one of the things that we do is we build kind of ways to identify what kind of phenotypes of curves and data is one that like leads to discordance and which ones lead to concordance. Like everyone sees it the same way. And then there's these types of tricky figures or curves that lead to discordance. And then what do you do with that? Like I think the wrong thing to do would be to say, for those, we're only going to trust like this one or this other one. What we're trying to do is figure out how to like, what are scientists seeing that we can teach. All right. And I think that's it's a small microcosm, but I think that thinking is what I'd love to see across the industry to benefit from both human and artificial intelligence capabilities.
I love that. And I think it's super important, but and I think it's important to have executives like you that are leading that mindset. Because what you see at some of these companies, you've got some people from, generations past that sort of blockade some of the new information. And you have to kind of really convince them, hey, why don't we try this? So this balance needs to be had, I think.
Absolutely.
Yeah,
it wasn't long ago that we had that kind of challenge with maybe automation, right? Like a decade ago, that was the kind of what played out with just using lab automation. Today, it's a different technology. That's a natural tension.
Yeah, a robot in your facility is no longer a.
What is that?
The pipetter's fine.
Oh my God, I don't know how many hours I spend convincing people we need robots, and now we have to do it with AI.
It happens in every industry, but I think it's a really, really great way to start wrapping up on that. I think that really punctuates it quite nicely. And we have our favorite segment that some of us do and do not participate in, but it's always our guest has the honors of our pick of the week. It could be anything, something in AI, a biotech paper, a tool. We've heard about some of it's funny, picks of the week have become later on shows, with people that work there. So, if there's a tool, we might end up interviewing the creator of that tool. You never know that's happening. Okay. Or a podcast or a book or something you've been listening to or reading that's we should check out.
I have a couple of little things that a couple from this week and one that is on my mind. A couple days ago, there was the FDA released a AI-based kind of reviewer called ELSA. I think it's meant to review applications, and I think it's medical devices based, to try to kind of identify and predict like reviewer comments or areas of further investigation. It's early and it's unclear how well it's going to work in its current iteration, but I love that idea. I love that like trying to inject AI and part of the approval and regulatory process that's really challenging and difficult and oftentimes like bottlenecked by timelines and complicated. So this is a very recent thing. I'm kind of curious to monitor it moving forward. And on the other end, there was this company called Future House. They released, I think, a new model of their AI scientist, a chemist, that I've been really interested in kind of understanding a bit better. Also, still kind of early days on that, but I'm curious to see more of that and understand how to kind of plug that in to our existing workflows and amplify our scientists.
Same, excited to see that update for sure.
Yeah.
Awesome.
Can I use his as mine? That last one.
One of these days, you can use your PlayStation 5 as your pick of the week.
Oh.
No, we won't let that one rest. I don't know what he. No, besides that, no, okay. I don't have. I realized I put it in my luggage for my next trip, but it's just a little thing I got while I was on my last trip in Germany. It's called the Plug Bug from a company. I believe they're still based in South Carolina. That's the name. Their name comes from that. It's 12 South. The spelled out 12 and the word South. And they are most often found in the Apple Store, but they make something called the Plug Bug, and it actually has a Find My built into it. So for those of us that lose chargers, and it's a nice backup because I was like, oh, I don't have a way to find my luggage. You know, these days, like an air tag or something like that, it's pretty common you put it in your luggage. But I had this piece of luggage. I'm like, I don't have anything to put in it. So I'm like, I'll just put this thing in that bag. And there it was. I could see it, sitting on the tarmac at the Frankfurt airport. I'm like, oh, yeah, cool. Works. Nice. So it's dual purpose, right? You get about 100 watts of charging, 2 USB-C ports, but also you can use it as your luggage finding tag.
So it's like a universal plug?
Yeah, it's just a plug. It has switchable. It doesn't have like the universal plug thing. I think those tend to break down. This actually has a whole thing of different plugs, UK, Korea, US, North America, all that.
Sweet.
And you just slide it in. And, it's like there's definitely a plug that was designed for travel. Somebody really put the thought in there. And boy, that thing charged my iPad in a matter of, it was fast. I had 0, went to 100%, I think, in, 30 minutes or something. So
wow.
It's got a lot of power to it. But Plug Bug from 12 South. There's like 2 models. There's a high power model and a low power model. That's my pick of the week. No, just
I'm buying that.
Yet another gadget from me. I tend to.
Those are arguably more useful.
That's right. And but thank you so much for joining us, Charbel, and it's been really fantastic talking with you. And for listeners out there, you can check out Charbel on LinkedIn as well at Charbel Eid, and also check out BigHat Biosciences at, you guessed it, BigHatBio.com. You can check it out. He is on the website and direct, VP of platform there, and so super excited to see what the future holds and what's coming next from BigHat Biosciences and all the work you're doing over there. So thank you so much for joining us. And for our listeners out there, if you'd like to hear more about how AI is transforming the industry, check out our Thought Leaders series. We've got a whole series up there now on YouTube. Sura has been just taking it to the next level on the shorts. So if you want quick previews of things, you can go to youtube.com at Helical Brew. And we'd love to hear from you. There's ways to reach out to us, of course. Comment section is not locked on our videos, so feel free to reach out in the comment section. You can hit the like. Maybe. No, just kidding. You can go to our YouTube channel or engage with us directly at helicalbrew@gmail.com. We'd love to hear from you and have that engagement from our viewers and listeners. Always available, always a little bit behind, I'll admit, in recent episodes. There's the iTunes and Spotify podcast approach if you like sort of the more passive way that they'll automatically populate on your phone, or YouTube if you want to go listen on that. With that, Charbel, thank you so much.
Thank you so much for having me. This was great.
Pleasure. And this episode of Helical Brew is a wrap.
Thank you. All right.
I thought you were going to say brood.
Thank you for joining us on the Helical Brew. We're glad to have you with us. For more episodes, please visit our website at helicalbrew.com. That's helicalbrew.com.